introduce Dr. Josephine Wee. Dr. Wee has been an assistant professor in food science at Penn State since 2018. Before joining our community, she obtained a bachelor's in food science and a dual title PhD in food science and environmental toxicology from Michigan State University. Following her PhD, she was a postdoctoral fellow at Cornell University, where she studied yeast evolutionary biology, and she also had a postdoc position at Michigan State University in nutritional immunology and toxicology. Here at Penn State, her lab group uses molecular biology and genomics to understand the role of fungi in food safety, quality, and sustainability, including working on developing fungal mycelium biomaterials, fungal degradation of plastics, and generating novel food proteins. And of course, exploring fermented food microbiomes, which she'll talk about today. So please join me in welcoming Dr. Wee.
Thank you. Good morning, everyone. Can you hear me okay in the back? Awesome. Thank you. All right. Thank you so much for being here today. My name is Josephine Wee. As Nicole mentioned, I'm an assistant professor in food science. I've been trying to convince my mom that I don't assist a professor, so I feel like they can get rid of these titles. She still thinks I assist a professor, but I'm like, I'm my own person. Our team is also one of the recipients for the Interdisciplinary Innovation Fellowships from last year that really gave birth to this project that I will share with you today that really spearheaded that. So I wanted to extend my gratitude to the center and its community for all the support so far. So I'm really excited to share with you an ongoing project in our group on how we are leveraging fermented food microbiomes, specifically sourdough microbiomes, to optimize an ancient fermentation process. So our vision is that by understanding the functional characterization of these microbiomes, we can kind of tailor these simple, I'll show you why they're simple to us compared to other ecosystems, tailor these simple and powerful systems to address some of the greatest challenges when we think about our food supply. And I threw in a word there, sustainable. From our lens, sustainable means safety, quality, cost, and accessibility, as well as nutrition. Okay, so that's the context of sustainability that I want to focus on. Nicole, do we need to check whether they can hear us from the chat? Oh, was that you?
Yeah, that's me messaging them. So we're good. Thanks.
More advanced. Okay, so I'm originally from Malaysia. At age 19, I left my hometown. Just a little background about myself, my journey here to Penn State. Really in pursuit of education and knowledge in America. It's kind of my dad was a chemist. He did, I didn't want to be a chemist. I really wanted to be a chef. I love food, but dad was like, no, you're going into chemistry. Okay. So I was like, so food science became like that sweet spot for me that I could convince him of why I wanted to do science. I received my bachelor's of food science and PhD in dual title in food science and environmental toxicology from Michigan State University. In my PhD, I became really fascinated with sort of the transcriptional regulation of secondary metabolism. A secondary metabolite that I worked on was aflatoxin. At that time, I became really interested as to why a fungus would produce these toxins. So that was my background, sort of in the kind of the promoter regions driving gene expression. And then I was kind of like, okay, talking about the bad fungi, right? The evil fungi. I'm like, okay, maybe I should go kind of to the good side. So I was like, okay, maybe I should learn about yeast biology. That's where I found sort of really this nice spot transitioning into yeast biology, where I did a postdoc at Cornell. I became kind of obsessed with then the non-coding genome of yeast. So then I worked on kind of RNA decay and trying to understand regulation from a decay point of view. And then because of immigration stuff, had to take a short, like 3-month postdoc back at Michigan State before I transitioned to my position here. So my lab really kind of bridges all these loves that I have, the fascination for filamentous fungi, yeast, as well as a little bit today on nutritional immunology, the project that I'm going to share with you today here. So I started here in 2018. Our team is broadly interested in this interaction between genotype and phenotype. Really, what are the molecular mechanisms that are driving phenotypes in fungi? And we ask this biological question really from the context of food mycology, which is the study of food-associated fungi. The story I'm going to share with you today, which is the third story here, the third project, involves the characterization of a fermented food microbiome, predominantly sourdough. We're really asking the question, can we leverage the sourdough microbiome to control and alter biochemical function? Okay, and I'll explain more as to what biochemical function means. I thought I'd put this two here, two other projects that are going on in the lab with the mycotoxins and food safety. We're trying to prevent and reduce mycotoxin in our food supply, as well as exploring the creation of fungal biomaterials for use in food, proteins, as well as biomedical scaffolds. And I'm a huge champion and fan of interdisciplinary work, so I, of course, welcome any collaborations and opportunities to connect. Okay, so I believe I'm the first speaker let me try to see if I can move this. Just click it.
So, yeah, click that.
I mean, if you just move your mouse away from it, it'll go back.
Click on the dots, click on the ellipsis, the three dots. Yep, there should be hide floating controls.
Oh, yeah.
Second line, yes.
There you go.
There?
Yep.
Yes. Thank you.
Bless you.
Thank you. We need to sleep on this light. Yeah. Now I need to find the mouse. Okay, hold on. Okay, got it. Perfect. Okay, so I believe I'm the first speaker this year in the microbiome series that's really talking and discussing about fermented food microbiome. So I thought I'd take some time to really go through, give you a brief overview of food fermentation, and particularly the production of leavened bread. So the concept of bread production dates back to the 3000 BC, with evidence that you see here in tombs in Egypt. Okay, can anyone tell me what they see here? Yeah.
Systems of production.
Yeah, production. It's the first evidence of this process that goes into sourdough. So paintings that date further back just capture a single product, but this is one of the first evidence capturing kind of the process of harvesting of a raw ingredient, in this case wheat, and then the transformation of that raw ingredient into a final food product, which is bread. I'm not sure if you see those little loaves up there. Okay, so that's pretty cool. And then there are writings from Pliny the Elder in about 770 AD that recounts this observation of skimming off foam from beer and baking bread with it, and the observation that these breads had a lighter texture. So that was the first record of use of yeast in leavened bread. Okay, so that sets context for some of the early references to a technique that we use today called backslopping. So taking skim foam off beer, adding it to yeast. So that's a technique called backslopping, which is important in sourdough because you skim off part of the flour, yeast mixture, and add it to your next batch of dough. Okay. So that's still used today in sourdough production. Okay, so the concept of sourdough fermentation is pretty simple. Upon mixing flour and water, there are wild yeast and lactobacilli that are present in the flour that are endogenous to the flour mixture that form this symbiotic community. And I know our microbial ecologists like to think about this as a microbial ecosystem. And this is what we call a sourdough starter culture. Okay. So I'll be using the term sourdough starter culture quite a lot today. This starter culture produces carbon dioxide. So these are the beautiful pockets that you see in your crumb, kind of when you cross section and cut them, as well as the taste. So organic acids that are produced when you eat a piece of sourdough, you kind of taste this tartness. You can feel it in your tongue. That's the production of lactic acid that comes from lactobacillus populations. Okay. And of course, there's a whole slew of other metabolites that I would share with you today as well. When you compare this to conventional dough, which is made from commercial strains of the yeast Saccharomyces cerevisiae, also known as baker's yeast, you can sort of see this comparison. Baker's yeast really rose to popularity. Does anyone know which century? With the Industrial Revolution, 19th century, around that time, because we wanted really quick ways to ferment, rapid ways. So think about your Fleischmann rapid rice yeast or your active dry yeast. So we really wanted a way to make, really quick fermentation time and as well as consistent breads, right? So just look at the cross section of this loaves. Okay, of course, sourdough fermentation, as you saw, has persisted for centuries. But I would say that its true resurgence really occurred during the COVID-19 pandemic in 2020. There were reports of flour shortages all across the US, and how to make sourdough bread was the most Googled recipe of the year. So the interest in sourdough as a pastime was obvious, right? People with a lot of time in their hands. But, why are consumers interested in sourdough? So first off, the interest in fermented foods has generally increased over the last couple of decades. If you followed the work of Justin Sonnenberg, for example, on linking fermented foods with the gut microbiome, and sourdough as a fermented food is part of this craze too, okay? So with particular to sourdough, there are a lot of people that report insensitivities to eating sourdough that we don't, they kind of claim that when they eat sourdough, they feel fine, but when they eat conventional breads, they feel bloated and sort of this kind of anecdotal recollection or reflection, okay? So, so first off, the interest is really this idea that it's better for my health. Okay, so I've heard that fermented foods are good for you. I feel better when I eat sourdough compared to other breads. And the second is this perception of simplicity, okay? So what you see here are two ingredient labels, all right? The top one is Trader Joe's organic sourdough. And the bottom one, I'll save the name for now, but can someone tell me what is your first impression on these ingredient labels?
It's long and has complicated names.
Yeah, so the bottom ingredient label, which is the ingredient label that you find on your beautiful Wonder Bread in the store, the ingredient labels are long and you don't understand a lot of these names, okay? So the other interest as to why people or consumers like sourdough is this connection to the simplicity. I can make it at home, so the use of simpler ingredients that they think are better for them. And it looks like sourdough breads have fewer ingredients on the label, okay?
Question.
Yeah, go ahead.
How do companies keep consistent with sourdough formulations if it's supposed to be a wild culture?
Yeah, so that's a great question, and it depends on the commercial company. So there are three types of sourdoughs. There are sourdoughs that you see that actually add lactic acid into it. So it doesn't matter, it's just a wild ferment, then they add commercial yeast to it. So you get kind of the best of both worlds, a little bit of the wild culture and then the commercial. And then there are type 2s and 3s that have a very limited, like selected for population. So it's beyond your 3 strains of commercial Cerevisiae. You may have 3 lactobacillus and 3, but essentially it's backslop, it's not controlled in that sense. So it depends on the company. Most large scale companies that make sourdough actually have the process down to a quite controlled stage where it's a combination of strains that they put together, that they select for, or they mix essentially. But the traditional like ancient sourdough, very few companies use it. You might get artisanal bakeries that use it. Really for that reason, right? It's hard to control, it's long fermentation time. So there's really no buy-in from companies. Yeah. Did you have a question, Seth? Or was it there?
Facilitating.
Yeah, okay. So now let's take a deeper dive into the science of sourdough microbiomes and what we know. Okay, So these in the kind of the early 2005, a group in Germany really started asking what is the molecular taxonomy and genetics of sourdough, and at that time they focused on the lactic acid bacteria. So that was kind of this huge interest in lactic acid bacteria, really from the probiotic movement as well. So this was the first glimpse using RAPD. Does anyone know what RAPD is? So it's basically a traditional PCR-based methods where you make primers that amplify multiple regions in the genome, and you kind of get this fingerprint. Okay, so the take home here is that we see a lot of, at the genus level, lactobacillus, quite diverse in the sense of its species as well. But what's more interesting to me is the diversity in the strains that are present here. Okay, so that was the first glimpse into who was there. And then about 10 years later, Mike Gunzel from, I believe he's in Canada, in Alberta, really started asking, what is the composition and function of this sourdough microbiota? So I'm going to summarize that for you, and our microbial ecologists like to think about these. So this idea of, are these sourdough microbiomes going through diversification? Are they drifting? Are they, widely dispersed? Is it one species that is particularly fit that kind of outgrows, the others that are present? Okay. So it's a really complex question, but I really like his idea and his concept of these four kind of microbial ecosystem types that are working at a given time. Okay. And then in 2017, the views from Belgium, I believe, started linking microbial ecology to the process technology, starting to ask, what are the function of these microbial communities? Okay, this is my favorite part of this story. In 2018 to 2020 is when we experienced sort of this paradigm shift in sourdough microbiome, where Rob Dunn from NC State and his team really asked this question: where do microbial species come from? Where do these microbial species come from? Any thoughts? Anyone? Where do you think your sourdough communities come from? Predominantly.
My cats.
Your cat, okay. The environment or animals, okay. Good guess. Okay, so what happened in this 2020 paper by Rees et al. From Rob Dunn's group is that they chose 18 bakers, I believe, that were in America and in Europe. They asked them to bake breads with the same recipe, with the same protocol, essentially, but with their different sourdough microbiomes, okay, sourdough starter cultures. So what you're seeing here is the kind of the take-home message from this is that the ingredients in sourdough is where predominantly the microbes come from. So your flour and your water. A little bit from the hands of the baker, but majority of your microbial species or diversity really comes from your flour and your ingredients, okay? So Rob Dunn and his team did not stop there. They felt like there were a lot of questions that were still left unanswered on, kind of how does the microbiome vary depending on geography, depending on location, depending on parameters such as water, where the baker, how it was stored, for example. So this study really set the groundwork for the Global Sourdough Project. Okay. So what happened in that Global Sourdough Project is that Rob Dunn's group teamed up with Ben Wolf from Tufts University, as well as some of the other collaborators, to really ask this question, how does each factor, people, water, flour, climate, contributes to the sourdough microbiome? Okay, in this project, they recruited 560 bakers from all around the world who sent essentially their sourdough samples to Tufts University for sequencing. Okay. So one of the lead authors, Anne Madden, so this is published, Landis et al., and we'll go through what you see here in a little bit. This is really the largest collection to date of a fermented food microbiome. So, it makes me really happy that Ben Wolf and Rob Dunn really did this and created a really wonderful resource for us to study. Okay, so what this study showed was that Saccharomyces cerevisiae, so if you look at the yeast, kind of this soil color, brown color, Saccharomyces cerevisiae dominated these communities, but you still see some diversity in the fungi that were not really studied beyond the LABs at that time. When you look at the bacteria populations, LABs, which are the lactic acid bacteria and the acetic acid bacteria, are what dominated these communities. So about 97% of these communities are LABs and AABs, but still a lot of diversity that exists. Okay, so initially when I shared that these are pretty simple communities to study, is because that they, around 20 fungi, 20 fungal genus, and 5 to 6 bacterial populations, which makes it really tractable to study. Okay, so despite the presence of this diversity that I talked about across individual sourdough samples, there are certain taxa that Ben Wolf observed that were based on geography, climate, as well as processing. Okay, so on your left here, you see clusters of microbes based on geography. And then on my right, certain microbes are associated with process parameters. Okay, so for example, the age of the starter, whether the starters are maintained using whole wheat versus other types of wheat. Okay, and whether it was handed down. So one of the things with sourdough is that it was handed down from my grandmother, for example, for hundreds of years. Okay, so some of these populations stayed over time. Okay, so what they did was also studied a couple of parameters related to food processing. So this is a really busy slide here, but basically the take home is that community composition really correlated with volatile organic compounds, which the team found the acidic acid bacterial populations was what was contributing to this kind of change here. Okay, so this functional traits that you see here, that a lot of things are going on, was contributed by acidic acid bacteria communities. Okay, so I think it was 2021 now, so September 2021, I get an email from a friend, Charlene Van Buiten at Colorado State University. She said, I have a grad student that's really interested in sourdough. Do you want to be part of it? We don't know how to go about it from a microbiology point of view. Are you interested? So I was like, okay, so let's talk. So Charlene was really interested in how does sourdough microbiomes affect the quality of sourdough breads. Okay, so Charlene's expertise is related to gluten immunogenicity, and I'll share with you a little bit towards the end on that. And so really we looked at this project as 2 arms, which is the structure and function arm on the left here, with linking community composition and the microbiome to physical chemical properties of bread. And then with Charlene's side, extending to the nutrition and health side, linking really structure to function. Okay, so that's kind of our framework. Again, asking this question, can fermented food microbiomes be leveraged for food safety, food quality, as well as nutrition? Okay, and of course, how do we define quality? Okay, so from our perspective, we can break that down to 3 sort of categories where scientists would like to reduce everything down, which is why microbiome research is so hard for me, because it's everywhere and so complex. So anyway, we define quality using these 3 categories, okay? So the first are things that are noticed by the consumer when you pick up a bread, its texture, appearance, flavor, and aroma, okay? And then on the other side, our categories related to shelf life and bread stability, okay? So then you get into the quality and safety aspects. And of course, in the middle lies this category where we're asking about how quality relates to health and nutrition indicators and outcomes. I should also mention that about 1/3 of the bread that is manufactured today ends up being thrown away because of shelf life issues, okay? So we're hoping that there may be a reason to move back to more complex communities to bake breads, so that we can extend its shelf life as well. So that's kind of the food safety and sustainability aspect. Okay, so our workflow for addressing quality looks like this. So at that time, I told Charlene, I was like, I know of Ben Wolf's work, we've met at conferences. Why don't we just take some of his cultures and do an initial preliminary study? So what we did was with Ben's help, we selected 20 initial sourdough microbiomes that best represented the diversity. Okay, so remember that 500 kind of all over the place diversity, Ben selected what represented the most diverse communities for us. Okay, so we got 20 sourdough cultures from them. So we already have sequencing data from Ben's study. We fermented the doughs because with sourdough, you need to let it go kind of a long fermentation, 24 hours. So we can talk about how that variation affects as well. And then we use those ferments or sourdough starter cultures to bake breads. Okay. So for the physical and chemical analysis, we compared these factors such as like crumb compressibility. So all of your texture, what your consumer is looking for. So crust color, water activity. We use ICP-MS to measure differences in mineral profiles. So these are your elementary minerals, kind of getting to that nutrition aspect. We also did some really cool metabolomics in collaboration with Jessica Prenny, which I'll share with you a little bit about her work as well, to measure the metabolomics as well as some preliminary cell culture work. Okay, so these, this is a snapshot of, this is not our data, this is Ben's data. We basically, used this data, got hold of these 20 samples that you saw here, worked through the pipeline again to make sure that we can repeat this. Another thing we're doing in the lab right now with Ashley Ostrom, which is my grad student, we're resequencing these because we don't know how much they've drifted over time. So we have questions on storage as well as passaging. So we have 20 of these sourdough samples that represent fungal and bacterial diversity. Okay. So if you look on your left here, you see the fungal communities are much more than the bacterial communities. So about 17, I would say, genus represented, and with bacteria, about 5. Okay, so this is really nice for us to really use this system. Okay, this is just another way to look at the data. And again, I don't come from a microbial ecologist perspective. I just try to make sense of the data from a biochemical point of view and a microbiology point of view. So what we're seeing here is that there's a greater fungal diversity compared to bacterial diversity, just with this 20 samples, okay? I know it's a small sample size. We're working on expanding this, but bottom line is that, we see greater fungal diversity, but does this fungal diversity really correlate to function? That's what we're trying to understand. Okay, so again, another way to look at the data, this sort of hierarchical clustering of samples reveals similarity between these samples. So the reason why we did this hierarchical clustering is to understand whether samples kind of cluster together based on the bacterial communities, so we can use that as a representation of downstream metabolomics, for example, or downstream proteomics, okay? So we see here that there are similarities in samples, but the clustering of the samples don't necessarily line up when you take bacteria and fungal populations together, okay? We're still trying to make sense of what this means from a functional perspective. Okay, so now let's look at how some of these sourdough microbiomes affect physical and chemical properties of the sourdough bread. So to address this, we measured a host of parameters that I shared with you that consumers care about in breads. Okay, so we measured acidity, water activity, your different production of organic acids, low volume, compressibility, and color difference. Okay, so we found that these unique sourdough microbiomes influence each of these parameters. While the difference existed across these strains, what's really cool to us is to see this clear separation between yeast dough compared to the sourdough microbiomes. Okay. So we see subtle difference across samples depending on which parameter we look at, but it was really nice to see this really clear separation that gave us sort of a control, essentially. Okay, we see this reflected when we cluster these physical and chemical parameters of these cultures. Again, you see them really nicely separating with the control yeast control. And a principal component analysis showed us that what was really driving in terms of the physical and chemical properties, what was driving differences across these samples were the organic acids and the texture of these breads. Okay, so how bread looks and feel and tastes isn't the only factor when we consider when we think about sourdough breads. We're also interested in how these sourdough microbiomes affect the nutritional profile of these breads. So in collaboration with Jessica Prenny at Colorado State University, she is partially funded by the Periodic Table of Food Initiative. I'm not sure if anyone knows, it's a Rockefeller Foundation project that essentially their vision is to, just like we want to sequence the whole supply chain, they want to get the proteomics of the entire, basically the food supply. Okay, so it's a really cool initiative. So she got funding to do some of this work that I will share with you from the Rockefeller Foundation. Okay, so when we looked at the elements of these 20 sourdoughs, we really don't see any correlation with these 26 identified elements between bacteria and fungal communities. And a preliminary glimpse into the metabolomic profile is more interesting. So there are about 65 known and unknown compounds, which allowed us to classify these 20 microbiomes into 3 groups that you see here in green, blue, and red. Okay, the top is the clustering of the samples, so the microbiome samples, and then the bottom on your right is the loading spot for it. Okay, so looking into what these metabolites are. These differences in metabolites are really driven by these groups of antioxidants, anti-inflammatory, and antibacterial compounds. So what we think is that these compounds, the presence of these metabolites, are what is impacting the nutritional profile of these breads. Okay? But also considering, for example, the acids that are present, it could also affect bread stability as well as the staling of these breads. Okay? And I believe the yeast sample was present in group 3, which is the blue group. And you also see that this group generally produces less of these metabolites. Okay. Okay, so I wanted to switch gears a little bit in this final section that really speaks to this idea of the One Health microbiome concept. Can we leverage the microbiomes of fermented food to alter gluten structure, gluten degradation, as well as its downstream inflammatory capacity? Okay, and this is a case study on gluten, but you essentially can look at it at, in any kind of allergenic proteins. Can fermentation be a way to really decrease allergenicity of our food ingredients in a way that perhaps want to sell more of it or make it more accessible to a population that has increased allergenic reactions? Okay, so this is gluten, all right? So when you kind of ferment a dough, when you pull it together, this is your idea of what gluten is. So gluten is the structure in bread that kind of binds it together. There's this string in the dough that you see that creates this wonderful texture when you cut into your bread. At the molecular level, gluten is a class of proteins that are made out of 2 subunits, gliadin and gluten. Gliadin kind of is proline rich, okay? It's kind of globular in that sense, and glutenin is more fibrous. And together, the interaction between these 2 subunits form your beautiful gluten network that you see in your breads. Okay. So what gluten does is it stimulates innate and adaptive immune responses when it comes to celiac disease. So for those of you that have celiac disease or know of someone, it's sometimes really rapid. Okay, so that's your innate response there, and also more long-term with your adaptive immune. Okay, and then there are some people that report non-celiac gluten insensitivity where they're not diagnosed with celiac, but by consuming wheat products, it makes them feel bloated. There's just no explanation for what they feel. Okay, so that is your sort of non-celiac gluten insensitivity. Okay, so it's not clinically diagnosed. Okay, so what we think is happening here is that really the motivation for this is this widespread belief that I shared with you, right? You can kind of insert like my friend or my family, my daughter can't eat gluten, but they can eat sourdough bread. Okay, so this is just anecdotal. If you kind of look into articles in New York Times and other kind of high profile news articles, there's this sort of anecdotal I wouldn't say evidence, but observation or accounts that people with insensitivities, consumers with insensitivities can consume sourdough bread, but not commercial bread, okay? So what we think is happening is that in your commercial bread, where gluten structure is intact, and you only have maybe 2 or 3 types of Cerevisiae, which is your baker's yeast, doesn't do much to the gluten structure, okay? So you get intact gluten structure and function. But when you let gluten over time in a fermentation of a complex sourdough microbiome, or simple, complex, in a sourdough microbiome, what happens is during that long fermentation time, the microbes actually modify the gluten structure in the bread, okay? So this is what we're hypothesizing is happening. So we did some initial studies to test this hypothesis. This was mostly done in Colorado State with Charlene's lab. We first looked at free amino acids, and we see that the different microbiomes, again, your x-axis are just the yeast control and your 20 sourdough microbiomes. We see differences in free amino acids measured by essentially giving us a glimpse into protein hydrolysis, okay? Okay, so free amino acids are pretty broad. What does that mean? So in order to understand the specific effects of gluten, what we did was we took different fractions of these sourdoughs and we compared these different fractions looking at high, medium, and low molecular weight gluten, okay? So what you're seeing here is essentially the samples that have higher low molecular weight, so meaning more chopped up proteins, which is the blue portion. You would suspect or hypothesize that these are the fractions that would not elicit an immune response downstream, okay, or to a reduced extent, okay? All right, so what you're looking for is an increase of these low molecular weight proteins, which is hydrolysis of these glutens, okay? All right, so Charlene is in the middle of doing some of these experiments with Keiko-2 cells. I don't have the data, but she shared with me that sample, I think 446, for example, and 35, we do see some impacts on intestinal barrier with Keiko-2 cells, which is pretty exciting, but I don't have the full set of data just yet, okay? Okay, so overall, these preliminary data showed our team that sourdough starter microbiomes are really powerful, right? They really impact the outcomes of the breads they produce. So about a year after that initial email from Charlene, we went to the USDA, NIFA, with all of these preliminary data, all the cool data that I shared with you. Last year, we wrote in September a NIFA grant that was funded this year to work on this project. So I think it was really tough in the sense if you kind of go back and say, what if I didn't answer that email, you know, or you know, what if I didn't just pull through? What if I said I'm not a microbial ecologist, I can't work on this, right? So this seminar really gave me a chance to kind of look back and look at all the things I said yes to and also said no to, right? So this was really nice. So what's next for us? So with this funding from USDA NIFA, we are working with Ashley Ostrom here, a grad student in my lab. We're really asking how are these stable over time? I guess to also Dr. Ganda's question on if this were to be commercialized, you know, what is the effect on passages and storage on the stability and function of this microbiomes? Remember, there's 2 sides to this structure function, right? It's the microbial structure, it's the bread structure, as well as its function, right? And does the stability of these microbiomes impact its function, okay? I'm curious to hear from you. Do you think these sourdough microbiomes will be stable over time and passage? No, okay, Nicole said, yeah.
I'm just wondering if, since the hands of the baker had an influence over the bread, and you indicated that the starter for the next came from skimming off the bread
From ancient
fermentation?
yeah.
Does it matter if the person does the maker matter, I guess?
We don't know, right? Based on that initial work from Rob Dunn, he showed that the hands of the baker does influence slightly. but majority of the microbiome comes from the origin, comes from the ingredients. Okay, and I think someone said their cat, right? So I think a home baker would be quite different than a commercial setting of these sourdoughs, right? So I think, you know, we have some ideas. What I think is that there will be a core microbiome that contributes to function that is quite resilient to perturbations, just kind of thinking of a gut model, for example. But then when there are extrinsic factors like temperature abuse or an introduction of a contaminant from flour, it might kind of perturb that system, but we don't know. We think that a core would be quite stable, and then there is this accessory microbiome that may or may not be as important when it comes to function, but we don't know. So we're happy to hear any suggestions from you as we kind of tackle this problem on diversity and abundance over time. Yeah, Nicole.
For this project, are you looking at how the community level changes, or are you looking at strain SNP?
Both. So we're looking at community changes as well as remember those samples we have, that 20 samples. So I think first community level, and then of course the SNP, especially when there are enzymes that can actually degrade gluten that are present in these glucose. So, yeah.
Two-part question. Do yeasts have analogous to a bacteriophage that could like go in and make a mess?
Not that I'm aware of.
Are you considering on the bacterial side of these cultures, the potential impact of bacteriophage introduction?
We're not considering it, but I've read a couple of papers, like I think Ben Wolf had a paper saying that are these fermented food microbiomes a reservoir for antimicrobial resistance transfers, for example, right? So I'm not familiar with that, but I think we're interested to look into it, especially these communities give us a chance to look into it. So that would be really cool to look at as well, yeah. So in addition to the time question, we hope that shotgun metagenomics will help us. So one of our hypotheses is that if we see varying levels of protein hydrolysis and gluten across these sourdoughs, the hypothesis would be that these communities contain microbes and activity that can then hydrolyze gluten. So that's our hypothesis here. So we hope to get into the function with this grant that we have. And then being mycologists and food scientists, we're also interested in how these microbiomes impact stability as well as shelf life of these breads. And the last part here is we're really excited to also look into how ancient wheat varieties can enhance the functional properties of sourdough microbiomes. Okay, so if you go into the literature on non-celiac gluten insensitivities, there is a lot of records that talk about ancient wheat varieties that did not trigger gluten sensitivity, sorry. So there's something that has changed over time with our selection for modern wheat that perhaps could contribute to kind of this heightened immune response in our food supply. So we're also interested to look into that with Ashley. Okay, so I hope that this preliminary data or this kind of framework showed you how we can leverage the functional properties of a food microbiome. These are simple and powerful systems that can address some of the greatest challenges we have today in our food supply, such as accessibility, cost, processing, nutrition, as well as safety. Okay, so we didn't do this work alone. Of course, we thank, of course, our funding sources and these wonderful people that all did the work and said yes at some point to us. So Charlene Van Buten's group does all the gluten and sort of the nutrition and health side. We work on the first part of the structure and function. And then, of course, PTFI, which is the Periodic Table of Food Initiative Group. And EVA is funded by the FFAR organization. So with that, I thank you for listening, and I'm happy to take any questions. Thank you for your time. Yeah, question.
Yeah, thank you for your talk. This is all really exciting, especially for people with gluten sensitivities. I'm wondering if you foresee this as a possible mechanism for other foods, not just bread, like that contain gluten.
Yeah, definitely. We're definitely trying to expand beyond breads. Dairy products are a huge thing, especially if you think of how much dairy products we're consuming in America, as well as allergens that come from the dairy environment. So those, I think, looking at wheat as well as dairy would be our, major focus, with fermentations. And it's really interesting, you brought that up, Carolee, sorry, just one more last thing. With soy sauce, for example, soy sauce is a fermented product. You have very low sensitivity when you think of products that are fermented. So we're trying to, it's pretty complex, but I know just kind of very broad associations. Yeah.
Yeah, so I'm curious about the source of microbes in wheat, and I don't know at this point what percentage of the wheat in the US is fumigated. So I know certainly when we ship it overseas, it gets fumigated, and it used to be fumigated all the time. And my understanding is the reason that we use sifters is because in ancient times, before fumigation, you had to sift out the bugs, right?
Yeah.
And now we still use it because we like the texture of the flour.
Mhm.
But I think there are still flours in the US that are fumigated with phosphine.
Yeah, I don't know. I'm not sure on that question. We haven't asked that question from a fumigation point of view. I'm thinking about, so it's the wheat germ that's fumigated, right?
I think it's
The flour as well treated?
I think, I don't remember, it's been a while since I've worked with people who are fumigating things, but I think it's both products.
Okay, we can look into it. Actually, we should kind of look it up to see. I'm not sure. I think we could get to the question from like the organic versus not
Right.
point of view.
I mean, I think it could change what microflora is
That
there,
are
right?
present,
So is it coming in from the kernels from the wheat field?
Yeah,
Versus is it coming in after
after
fumigation?
fumigation. Yeah, I'm wondering if the metadata from Ben was, because they collected like 300 different qualitative sort of parameters with these 500 cultures. We should look into the use of fumigation. Yeah, great point. Thank you. Yes.
Maybe it's related, but it seems like you're heading towards a path where you will generate lab-built microbiomes using sterilized ingredients in order to optimize some of the features of interest, whether it's taste,
Flavor, safety, yeah.
Safety, et cetera. Is that feasible? Is anybody in that space right now?
I don't think it's feasible. I think food ingredients are not sterile environments. So for example, we didn't in the lab, we didn't autoclave these flours, for example.
Of course, yeah.
Right, so these are in addition to the background flora, for example.
But eventually, could you sterilize all the ingredients and then start to play as an experimental biologist?
Yes, in the lab, of course, that's
Yeah.
what we're doing. So we're trying to leave one out. We don't want to go to a single strain. So there are a lot of studies that use a single strain, try to do therapeutics with single strains. We think that these microbes are powerful as a community.
Yeah.
So in the lab, we can definitely test out function, but I think my vision is that these would be commercial starters as a whole community versus a single strain.
Totally.
Yeah.
Question in the middle?
Yeah, so I was just thinking about if you are sterilizing your flour, right? Just because I don't know, does that change any of the nutritional availability within the flour so that when you're like inoculating it with, say, your primed mixture of fungus and bacteria, would that change that community at all as a result of maybe nutritional compositional changes?
Yeah, I think it definitely would. I mean, just thinking about your denaturation of some proteins in flour environment, it's mainly carbohydrates. So you're probably going to get like some sugars, carbohydrates that are affected by heat, but I still think you can use it as a baseline to study.
Or radiation sterilization?
That, yeah. Mhm. Yeah.
I was wondering how stable or does the nutritional composition or the metabolites of the bread itself change over time?
We don't know that yet. We have some glimpse from the metabolomics, but we have to look into, for example, like carbohydrates, fats, very little fats, mainly carbohydrates, some proteins, minerals are a big one. Micronutrients would be a big one. And especially with the organic acids, what I think from a biochemical function point of view, I think the diversity and abundance of these organic acids is what's causing the hydrolysis further. So these microbiomes have enzymes that can chop up gluten or degrade gluten, but I think that process is further enhanced with the presence of acids that are in the environment. So we're trying to test these individually together.
Question in the back? Do you have a question? Yeah.
Yeah, thank you so much, Josephine. Interesting. I'm really fascinated by the sort of anthropological story behind how these, sourdough strain communities change over time. And I'm wondering if you know, or if we know, if there exists today any remnants of those ancient sourdough starters, maybe
Yeah.
not from Egypt, but like.
Yes, actually, it's from Egypt. So I came across a Twitter post. It was 3,000 years ago. It was a tomb that had some of these sourdough. If you just look into Twitter, and they actually could revive it from the vessel, and actually bake bread with it. So it exists. The thing with me is that I'm always like, okay, what's your control? What do you use as a control? Being a microbiologist, I'm always thinking about those questions. But we are in contact with some people that found these ancient starters. We just haven't acquired them because we don't really know what would it tell us at this point. Yeah. But that cultural aspect is really interesting. And a lot of, I should point out, a lot of these studies came from Europe and America. But I suspect that sourdough microbiomes originated in Asia, Africa a long time. We just had no way to report them. So I don't want, for me, I think that sourdough is not like kind of a Western civilization type product. It also, there's just lack of records in Asia now, for example, in other continents. So I think the cultural aspect is really cool, especially when we want to look at the wheat varieties piece. I just don't know the right questions to ask yet, of course, reducing this. Yeah.
So is the difference in the diversity between the fungi and the bacteria really just a factor of the pH? And is that going to be true for most all fermented foods where you have a low pH?
Good point. When we look at the 20 sourdough starters in terms of like just macro level pH, right? Very crude pH measurements and analytical methods. We don't see a significant difference across the 20 sourdoughs. Of course, that's compared to the yeast, which is like neutral pH. Our communities hover around 5.2 to 5.6 pH. But do you think that minor change could still affect these communities?
I'm not necessarily thinking from between those 20 batches that you're
like
looking at, but I'm just
stability.
the fact that you only had 5 bacterial species that were really core and something like 14 or more for the fungi.
I don't know. I think it's a good question to look, or at least try to either control for or look into. You mean as how it affects stability, right, and passaging and storage?
Yeah, why don't you have more bacterial diversity there, I guess is my question.
I think it's largely driven by pH, because with acidic acid, for example, and lactic acid, it's kind of a competitive metabolite. You kind of eliminate a lot of other bacteria that are present, especially the spoilage microbes. And the yeast and fungi predominantly like lower. That's why I think they're more. Yeah, so you're right in that sense. Yeah.
So in your hierarchical clustering dendrograms, you had fungal and bacterial relationships. And
Yeah.
if you squinted your eyes, you could see there might be some parallel structure between those two trees.
Yeah, I don't know. Can you combine?
That was gonna be my suggestion.
I don't know how to do it.
We have out of the phylosymbiosis literature, we've developed some tests for topological congruencies between microbial communities
Okay,
and their hosts. And
Yeah.
we can apply this then to the fungi and bacterial.
that would be wonderful because
Like a p-value on.
because I had Ashley doing all kinds of random manual things with colors, but I just, it's really just,
So
like what we're
we'll
thinking.
follow up with you on that. And I guess my question biologically would be, are there known symbiotic interactions between the fungi and the bacteria in these communities that allow them to form those semi-consistent relations?
Yeah, so Ben tried to get into some of these initial ones with like a synthetic community.
Yeah.
But mainly looking at bacterial interactions. I think the fungi side is what we're hoping to contribute.
That's awesome. That's fruitful area. Thank you.
If I may add to that, you had a very beautiful clustering from your physiochemical and metabolite perspective.
Yeah.
I'm thinking the bacteria and the fungi, they're interacting, right? What if you combine that data, that might get you closer to a final product metabolite profile, and you think?
Yeah, I'm trying to do that. Yeah, I keep thinking of all these ways to combine the data, but I don't know whether it's the right thing to do or whether I'm overlooking or being biased. So yeah, we're definitely interested in looking at the interaction and how that contributes to function. Everything is like association now, and like Seth said, if I squint my eyes, like I could see 446 clustering with one, and but I don't want to make sort of this kind of premature like excitement of these clustering samples. Yeah, for now.
Well, I know we're at noon, but I have kind of like technical questions, like, do you have an oven in your lab? Or like, where are you baking these breads, or is that happening, you're just working with the culture?
Yeah, so we actually bake the breads. These were not baked here, but actually, we have a pilot plant that we can bake breads, and yeah, so we take the cultures and then make the breads and measure them.
And the cultures, are you keeping them just in the fridge? Or I'm thinking like, especially with the passages, things like humidity and temperature are gonna be a big.
Yeah, especially as scientists, we store these at -80. So that's the question Ashley's gonna get to. When you thaw and freeze these cultures, what's happening to the microbiome? So we're trying to get to some of these questions we often yeah.
So cool, thank you.
Yeah.
Let's thank Dr. Wee.